All right, welcome to today's explainer. We're going to take a totally fascinating tour of our cellular factory to see exactly how our genetic blueprint is copied and transported. We're looking at one of the most, if not the absolute most precise manufacturing processes in the known universe, and it is literally happening inside your own body right this very second. Let's get right into it.
Here's the roadmap for our tour today. First up, the master blueprint. 2. Unlocking the DNA helix. 3. The 5 steps of replication. 4. Meet the RNA messengers. 5. Mapping RNA locations. And finally, number 6, the code of life.
Starting with Section 1, the master blueprint. Inside the cellular vault.
Okay, so think of DNA as the ultimate master document. Locked safely away inside the cell's nucleus, this massive double-stranded helix holds the permanent code required to synthesize literally every single protein your body needs to function. Because it holds all your vital hereditary information, it never ever leaves the safety of the nucleus. It's strictly vault-bound.
Moving on to Section 2, unlocking the DNA helix, preparing for duplication.
Now, before this master blueprint can be copied, we run into a major problem. How do we actually open it up without destroying it? Well, notice this key structural detail. The two sides of the DNA ladder are held together by weak hydrogen bonds. Those inner connections, they're basically an intentional, brilliant design flaw. Compared to the super strong outer backbone of the molecule, these inner bonds are incredibly easy to break. This is absolutely essential so the molecule can be quickly pulled apart to access all that locked up information.
Section 3, the 5 steps of replication, the assembly line in motion.
So here is the main event. It's a highly coordinated, super strict 5-step sequence. First up, the double helix physically unwinds. Second, it unzips right down the middle by breaking those weak hydrogen bonds we just talked about. Third, each of those original DNA strands now serves as a template. Fourth, free nucleotides swoop in to build a complementary new strand. And finally, step 5, the whole process finishes with 2 perfectly identical DNA molecules.
This is basically the ultimate biological zipper in action. You can literally picture it unwinding. And right in the middle, as those weak bonds break, the physical unzipping exposes the inner chemical teeth of the DNA. It's exactly this exposure that allows the strand to act as a hyper-precise template for copying.
Now here's a golden rule you absolutely need to lock into your memory because it governs everything that happens next. When those new free nucleotides are building the complementary strands, they are strictly bound by this pairing. Adenine, or A, always pairs with thymine, T, and cytosine, C, always pairs with guanine, G. This complementary base pairing is the ultimate failsafe. It guarantees unwavering precision when creating the new DNA molecule.
So the end result of this whole factory process? 2 pristine, totally identical DNA molecules. But check out their makeup. Each new molecule is half old and half new. It contains 1 original strand from the template and 1 brand new complementary strand. It's a flawless system for perfectly preserving your genetic code for the next generation of cells.
But wait, this brings up a massive logistical hurdle. We've successfully copied the blueprint, sure, but since this giant DNA molecule is completely restricted to the nucleus, how on earth do these crucial instructions actually get out to the factory floor, the cytoplasm, to build the proteins?
Let's find out. In Section 4, meet the RNA messengers, the cellular couriers.
Enter RNA, or ribonucleic acid. Unlike our vault-bound DNA, RNA is this nimble, single-stranded molecule. It's built entirely for travel and rapid translation. It has a slightly different sugar backbone, using ribose instead of deoxyribose. And here is the dead giveaway. It completely swaps out the base thymine for a different base called uracil. So, pro tip, if you ever see uracil, you know for a fact you're looking at RNA.
Section 5. Mapping RNA locations. Spatial geography of the cell.
Let's talk geography, because we have 2 main players operating in entirely different areas. Think of messenger RNA, mRNA, as the commuter. It gets formed in the nucleus where it picks up the genetic message from the DNA and then it physically travels out to the ribosome. Transfer RNA, or tRNA, on the other hand, that's the local resident. It is located exclusively out in the cytoplasm, just stationed there, waiting to do its job.
Mapping this all out gives us a really clear spatial picture. You have the single-stranded commuter mRNA leaving the nucleus vault and heading straight down to the ribosome. And right there, waiting out in the cytoplasm, is the resident tRNA. Those tRNA molecules literally grab specific amino acids and bring them over to the ribosome to perfectly match the instructions that the mRNA just delivered. It's an incredibly coordinated cellular handoff.
All right, Section 6, the Code of Life, summarizing the master plan. Let's just quickly recap the major takeaways from our explainer today. DNA, that's your heavy-duty, double-stranded deoxyribose blueprint. It uses thymine and stays permanently locked in the nucleus. RNA, that's your nimble, single-stranded ribose messenger and transfer molecule. It uses uracil and handles all the logistics out in the rest of the cell.
And why does all this matter? Because it's this elegant, multi-stage dance. From the mechanical unzipping of DNA to the spatial commute of mRNA out of the nucleus, to meeting tRNA at the ribosome out in the cytoplasm, that ultimately links amino acids together. This is exactly what forms the specific proteins that keep you alive, functioning, and growing every single day.
Which leaves us with a pretty wild final thought to ponder. Given the immense, rapid mechanical precision of this microscopic factory, what happens to the entire organism if just a single chemical tooth on that zipper gets mismatched during replication? Literally just one single error in the blueprint changes the messenger, which changes the amino acid, which can completely alter the final protein. It's honestly mind-blowing that this process works flawlessly billions of times a day. Thank you so much for joining me on this explainer, and keep questioning the incredible mechanics of life.